So let's dive right into what I call the ultimate exam room mystery. Picture this, right? You've spent months, literally months, memorizing every single detail of your textbook. You know exactly how a cell works inside and out. But then results day rolls around, and somehow brilliant biology students still fall short of that elusive A star. Like, why on earth does that happen? Well, welcome to this explainer. Today, we're going to completely crack the code of the Cambridge Biology 9700 syllabus and figure out exactly how to bridge that frustrating gap. You see, the answer isn't really about what you know. It's all about how you present it. The two biggest villains sitting with you in that exam room, well, I call them contextual blindness and the dreaded command word trap. Look, you might know the deepest, most complex biological mechanisms, but if you just sit there and list facts without applying them to the specific living scenario the examiner actually gave you, yeah, you're going to lose easy marks. And this quote right here, it brilliantly illustrates the stakes. This is straight from the principal examiner report. Candidates consistently confuse key terminology. Always define terms before using them in extended responses. Basically, examiners do not want generic textbook descriptions. No way. They demand absolute precision. They're hunting for that specific terminology that proves you actually understand how to apply these biological principles in the real world. So let's look at exactly how we can deliver that for them.
So, here's our roadmap for today. We've got the Highmark framework, core cell and transport strategies, mastering respiration data, genetics and bioinformatics tactics, and finally, your final examiner checklist.
All right. Section 1, the Highmark framework. Let's get right into the ultimate foundation for securing what examiners love to call your method marks. Think of this 4-step sequence as your universal toolkit for literally any complex question. Step 1, concept. You have to identify the underlying biological principle. Step 2, evidence. Pull those specific data points right out of the stimulus. Step 3 is reasoning. This is where you connect your evidence back to the concept using rock-solid causal logic. And step 4, wording. Refine it all using that syllabus-specific terminology. Using this forces you to rely on evidence-based logic, which totally transforms a superficial answer into a highly analytical one. Moving on to Section 2, core cell and transport strategies. Let's actually apply this framework to a classic assessment trap that catches out, honestly, thousands of students every single year. Okay, before we even peek at the data in a Benedict's test question, you absolutely must define the chemical process at play. We aren't just looking at a pretty color change here. We're looking at the specific reduction of copper 2+ ions to copper 1+ by a reducing sugar. If you just write down something like a reducing thing, you've already lost the examiner. Save the word thing for horror movies, right? Not your A-level biology paper. Now, notice how the cause and effect trap plays out in this flawed answer approach. It's so common to see students disastrously reverse the relationship between sugar concentration and color change. Or they just get hit with that contextual blindness we talked about, throwing down generic answers that completely ignore the specific experiment in front of them. I know time pressure in the exam pushes you to write super fast, but remember, precision is what actually scores the points. Let's just contrast these two approaches real quick. The flawed answer? It literally says, the color changed because it's a reducing thing. Ouch, that scores a big fat 0. But the improved top band answer actively extracts specific units and precise chemistry. It says reduction of Cu2+ ions to Cu+, producing 0.5 mole Dm-3. See the difference? It names the exact ions, explains the reduction process, and actually uses the precise data units straight from the experiment. Let's pivot for a second to another massive, high-yield area where precision is literally everything: managing carbon dioxide transport. The chloride shift is one of those topics that requires a really deep understanding of dynamic biological movement. Specifically, we're talking about the movement of chloride ions into red blood cells. And why? To maintain electrochemical neutrality during carbon dioxide transport. Systematically applying these core concepts is how you safely dodge the examiner's red flags for being vague. Notice how the top band approach works here. It doesn't just weakly say salt moves. Instead, it systematically links the formation of hydrogen carbonate ions by carbonic anhydrase directly to the hemoglobin buffering process, and then finally to the inward movement of those chloride ions. That kind of cross-topic connection, that's exactly what pushes a solid B grade straight up to an A star. All right, Section 3, Mastering Respiration Data. This topic right here often accounts for a massive 15% of your marks. So yeah, it's an absolute must-master area. The most frequent mistake I see here, relying on generalized textbook answers that completely lack the biological context of the actual data provided. Listen to me on this one. Making energy is the ultimate forbidden phrase. Seriously. Cells aren't magicians pulling energy out of a hat, right? They synthesize ATP. Let's see exactly how this plays out in a realistic exam scenario. Looking at this detailed breakdown of lactate fermentation, notice how the improved strategy totally ditches the vague word energy and replaces it with the quantified phrase net gain of 2 ATP. It also gets super specific, stating that pyruvate acts as a hydrogen acceptor and is reduced by the enzyme lactate dehydrogenase. By naming the specific chemical process, the exact enzyme, and actually quantifying the ATP, you are proving to the examiner that you have absolute mastery of the syllabus.
Now, the stakes escalate even further when you're dealing with complex structural diagrams like the electron transport chain. When you're staring down those intricate, multi-layered diagrams of membranes and proteins, you have to remember that the method marks are hidden deep, deep in the terminology details.
The flawed student approach completely ignores the experimental data and just leans on vague terms like energy production. It entirely fails to recognize the actual 3D biological reality of what's happening across that membrane. But the A-star strategy? It explicitly identifies the fundamental biological principle at work, chemiosmosis, and clearly details those precise proton gradients. So, the absolutely crucial takeaway here is that naming the exact chemical process, chemiosmosis, is literally what triggers the marks in the official scheme. Writing protons move to make energy is, frankly, a middle school answer. But writing chemiosmosis generates ATP as protons are pumped across the membrane? Now that is an A-level answer. It shows you truly understand the underlying mechanism of action. Okay, Section 4, Genetics, Bioinformatics, and Statistical Tactics. Let's tackle the final frontier of the syllabus, where higher-order mathematical and evaluative thinking aren't just helpful, they're absolutely mandatory. When you're exploring bioinformatics databases of nucleotide and amino acid sequences, you're going to encounter this context gap that examiners frequently and heavily penalize. Look, when you're evaluating these massive databases, you can't just list off features. You have to use comparative connectives, words like whereas, similarly, or in contrast. This highlights both the similarities and the differences in the sequence data. Ultimately, you have to prove you can evaluate the evidence, not just passively describe it. To flawlessly execute this strategy in data-heavy problems, selecting the precise statistical tool from this list is step 1. It's a systematic process. If you're comparing the difference between two means, you reach for the t-test. Categorical data, that's your chi-squared. Or maybe you're determining relationships between variables. Then you need a correlation test. Boom. Choosing the right tool right out of the gate immediately signals to the examiner that you possess advanced analytical skills. And finally, Section 5, the final examiner checklist. As we start to wrap up this explainer, we really need to consolidate all these strategies into a totally foolproof routine for when you sit down in that exam room. Looking at this overarching examiner's blueprint, I want you to basically burn the top 3 warning signs into your memory. Number 1, never ever confuse cause and effect. Number 2, never forget to check your units and double check your terminology. And number 3, always build cross-topic connections. For example, if a question is about cell structure, you should immediately start thinking about how that structure facilitates active transport or ATP synthesis. Connect the dots. You really want to memorize this final review checklist. Literally, before you turn to the next page of your exam paper, ask yourself, are my units verified and converted correctly? Did I define my syllabus-specific terms? Did I apply my knowledge directly to this specific data, or did I just sort of write out a generic textbook summary? Have I checked for alternative interpretations? Asking these questions is crucial because earning marks for your logical processes, even if your final calculation at the very end is slightly off, is the true secret to unlocking that A-star grade. So it turns out the mystery of the exam room really isn't so mysterious once you actually know how the examiner thinks. I want you to apply this 4-step framework to your very next past paper. And hey, let us know in the comments which challenging biological mystery or mechanism we should decode in our next explainer. Keep striving for that top band precision. You've absolutely got this.